Hello, students. Mr. Sherwin here, and I kind of have a fun biology video to show you. I wish I could share this with you live, like in person, but it just doesn't work out that way. It's it kind of goes on with DNA, and we've been talking the past few weeks of, DNA and how to replicate DNA, and then we went into this whole transcription and translation thing, where transcription was where inside the nucleus, the DNA kind of unzips a little bit, and messenger RNA will be copied from the DNA. Very simple, similar to how DNA copies itself, just being the one major difference. Instead of thymine, you now have uracil. And then the messenger RNA leaves the nucleus and it goes in the ribosome, clamps onto it, and starts reading that code that was created. That messenger RNA is a code. And each of these three letters, three amino acid sequences in the messenger RNA codes for a single amino acid. Well, say each of the three mRNA nucleotides code for a single amino acid. And then these amino acids kind of form and kind of chunk out of themselves out of the ribosome, and eventually it stops, and you got a long string of protein, and it comes up and it all folds up and it folds up and it gets transported somewhere, or it goes and does something. Well, today we're going to kind of look at that a bit more about what's really happening, a bit, maybe a bit more physical aspect of what happens to DNA when it comes to messenger RNA, to making a protein, and then what happens when that protein folds, which is kind of important too. And furthermore, what happens when something goes wrong? Because there are a lot of life forms on this planet, a lot of things that they're different because, well, something went wrong. And we're going to see like what that wrong could have been from, and when it could be really wrong, and when it could be kind of wrong, and just kind of what happens. And so it's pretty impressive, pretty impressive. So, and we're going to do this by, well, we're talking about the building blocks of life, right? So let's talk about this by using building blocks. Good old-fashioned Legos. All right, here on the screen, you should be able to see already, we have a DNA sequence right here. This is just, I've already broke this apart into our codons. Remember, the codon is the group of three here, the group of three. So CGA, TAA, TCA, TAA. You gotta see that that's what the kind of the codon actually is here. I already broke it apart for you. The one thing that I did not do is I did not put the start code, the AUG, or the end code. We're just skipping that for right now because we're more focused on that, how this egg's actually built. Now, if you remember from the, I guess you can see this a little bit, but you remember from the lesson we had on Thursday, that'd be Thursday the 6th, not 6th, Thursday the 4th of December, we talked about how can we decode this? How can we decode this? And we know that using the transcription process that's inside the nucleus, transcription inside the nucleus, that, my C was going to pair up with a G, and the G is going to pair up with the C. But the U, well, actually, the A is going to become U because there's no messenger RNA doesn't have a T. So, but the T becomes an A. And so we practiced this a little bit, and we got to the point, and I did this for you just to kind of save some time. This is the sequence. This is our sequence of messenger RNA, from our DNA. So we're going to say this is our main strand of DNA. We say, I don't know, it's a strand that codes for eye color. That's a fun one, eye color. So, and this is just one of many that code for eye color. So that's what this is right there. This is the DNA strand, and it makes this section of RNA right there. That's what the RNA makes. And as we know, RNA will code for amino acids. Messenger RNA codes for amino acids. And we can read that by looking at the code using the codon table that we talked about, either the chart or the wheel. And we can say that, okay, well, GCU stands for one amino acids and AAU stands for a different amino acid. Save a little time here. We'll look at a codon table in a little bit, but I went ahead and made that for you too. Here are your amino acids. Let's just go and see the whole thing. I also assigned a Lego for every single amino acid that we're working with. Notice we don't have all of our amino acids. There's quite a few more than this, but we're just going to use these right now to kind of show what's really going on here. So here's our sequence. We have alanine, isoleucine, serine, another isoleucine. We have valine, leucine, tryptophan, histidine, isoleucine, and aspartate, if I pronounce that right. But we have these. And these get produced each one in series. The first one, the second one, the third one, the fourth one, the fifth one, the sixth one, the seventh, keeps on going on and on. Well, one thing we talked about several times over this semester is in biology, structure equals function. Structure equals function. And for it to work, the structure of something that's created has to physically interact with something else. So we're going to say that for this eye color gene, it's going to have to physically act with something else in order to make the eye color actually come out, or maybe one small shade of eye color. And so in order to physically act with something, we have to have something for it to act on. So we're going to say that whatever the stuff is we're making, it's going to act on this thing right here. Call this a substrate. The substrate, it's just kind of a term in biology where I have one chemical react on another chemical, or in this case, one protein enzyme will react on another one. All right, so here's what's going to react on. So whatever we make is going to have to interact with this thing somehow to do something. Let's set you aside. You're fine. Now, proteins, you might have read that proteins fold. When this chain of protein here comes outside the ribosome, based on whether each individual of these amino acids like water or don't like water, hydrophilic or hydrophobic, they're going to fold in a specific way. And their final folding, when they finally fold, that determines their final shape. So this one we're going to kind of walk through it here, but it happens in sequence. So first, second, third, fourth, fifth, sixth, seventh, eighth, nine, and here's how it came up with. So we have, that's the wrong one. Here's the protein that we made at this. I'm not going to have time to build it in class, but we have the first one. There is our alanine. And then this kind of connected straight to the isoleucine, which connected straight to the serine, which connected to another isoleucine, which this one connected to, there is our valine right there. Make sure I'm on the camera. And that valine connected to the leucine connected to the tryptophan connected to the histone. I'm trying not to sing the bones of the body song here. The histidine connected to the isoleucine, and now the isoleucine here connected to the ASP. And so this is the protein that was made. It came out of the ribosome in this long chain. It all did its folding and everything, and now it's going to float around through the cell. And the way it's going to work is it's going to physically interact here. There's a spot right here, where this thing can interact with this substrate right there, and just kind of slides in very easily and fits just like a lock and a key. A lock and a key. It fits perfectly. I'm not going to push it in too hard, so I won't be able to take it out. But and now we can say this protein can now go interact with another protein, eventually come up with the eye color green or blue. We're just kind of playing with it. So that's how it works. That's how we have our DNA sequence becomes messenger RNA, which codes for amino acids. And then outside once the amino acids come out of the ribosome, they fold up and form some structure, and this structure goes and interacts with something else. And then other things happen. I say other things happen in a very generic sense because sometimes multiple steps have to happen for this other thing to happen. So that brings us up to, okay, that's great. I mean, we kind of talked about this a lot, but what happens when things go wrong? Why do we have redheads? Why doesn't everybody look the same? Why do we have, some animals, some flies might be albino. I joked around a few weeks ago, we talked about kneecaps eyeballs on kneecaps. Things can happen weird with DNA. And we got these things called mutations. Okay. And mutations, maybe they're good, maybe they're bad. I don't know. They happen. So I want to show you how mutations can happen on a genetic level. This is tiny, 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 tiny. First off, the thing you need to know is that mutations are caused by a couple of different things. One thing, it could be damage. It could be damage. And so we got our DNA here, our sequence of DNA, and some damage might actually happen to this and actually damages our DNA. It could be radiation. It could be sunlight. It could be some type of chemical exposure. It damages our DNA. If it damages our RNA, often what happens, because that RNA is just that small strip of nucleotide chains, your body just kind of tries to fold it. It can't do anything with it and just throws it away and recycles it. But if it damages the DNA, that could be bad because the DNA could duplicate itself. If I have bad DNA duplicating, cancer, it's a thing that can happen. But we're going to see here that, kind of what happens when we damage a small piece of DNA, what happens to the protein we're making. So our same sequence here, I'm going to take our players off the field here. Actually, we'll keep them there. Let's slide down a little bit. Let's say we're going to make one tiny change. Look at that cysteine right here. We're going to say that something happened. Maybe you got a little bit too much sunburn. Something happened, a little bit too much sunburn. And this alanine that was here in the fourth codon now suddenly changed to a cysteine. Something happened. Maybe when the DNA was duplicating it, it forced the wrong one in. It could be multiple reasons, but somehow it's this code now goes from U to a C. Okay. Well, the question you ask yourself, think about it. Is this bad? And the answer is, I don't know. Let's find out. Let's think about it. So we already know that these first three of this chain, we have our alanine, isoleucine, and serine. They're going to be the same because these codes are the same. But when you translate these, there's our transcription. Remember, transcription making mRNA, and translation going from the mRNA to the amino acid. We now have serine. Okay, serine. So interesting, but we've serine, and then the rest of these are the same: valine, leucine, tryptophan, histidine, isoleucine, and aspartate. All these are the same, except for this one right there. But come on, that's just one letter. Can it be that bad? I don't know. Let's take a look and see what happens when you change. In this situation, it's not all situations, we changed one little letter in the codon to see what happens. Change that middle. And so what happened, I went ahead and built this again. Here is this, remember, here's our original structure here. Let me make that visible. Here's our original structure. And then here is our new structure that we now have serine and serine and the rest the same. And so I can kind of identify everything here again. So we have alanine, and then isoleucine, and then serine, and then instead of another isoleucine, it's another serine. Okay, this is the one that's different right here. Kind of compare these two. So what's different? And because that's different, it's wider. The new one's wider. So by this being wider, I don't know, is something bad going to happen? I don't know. I mean, this can technically still come in and it could technically still bond. It's a bit loose, maybe it slides back a bit, but maybe that's not going to hurt anything. Maybe this part here will bond to something, or this bonds to something. I don't know. There's no way of telling what's going to happen. It's going to take many such mutations to really kind of see what happens if this doesn't work. If this is kind of critical, maybe it does cause a bad mutation. So we don't know. But it does cause the protein not to work the way it's supposed to. So this could be detrimental. This could be beneficial. We don't know what this would do. But this is an example of a mutation that does keep the structure of the protein around. So let's take a look at another one here. I'm going to get rid of some of these. I got to get rid of our players here. Let's look at this other mutation here. Let me scroll down. So we're going to now change. I'll write this above here. This initially, we're doing a second mutation. This is what the original was here. We're going to do a new one here. We're going to change this T to an A. We're going to change, sorry, the T to an A. We're going to change, this initially was an A. This initially was CAA. And we're changing this A into a T. And we're going to see what happens. Nothing else changed. That means all the rest of our proteins are the same. And I want you to notice what amino acid was made there by changing just that last letter in the codon. Think to yourself, what amino acid was made? I got it written down right there. Valine. And look at the original amino acid. What was the original amino acid? The valine. Here you have an example of a mutation happening, but it just mutates the DNA and nothing else changes because that different code still codes for the same amino acid. If you take a moment when we're done this video here, take a look at the codon table. There's a lot like this. You can change that last letter quite often. It still is the same amino acid. That's kind of cool. Kind of cool. All right, let's have another one. Let's have another mutation here. Let me find another mutation here. Yep, I got some more here. How about if I can find it? How about this one? Let me get back on the screen here. This one is kind of interesting because, slide down just a bit more here. You can see in red what changed, but we went from CAA to CA. In this case, I deleted this. What happens with DNA just kind of gets, a chunk gets deleted. It doesn't replicate right. This is called the deletion. In this case right there, by deleting a nucleotide, it causes this thing called a frame shift, where the entire codon shifts this way, and we now have something completely different we're reading. So instead of having reading in this sequence right there, all of these letters shifted one spot over to the other side, and now it's coding for a completely different set of proteins. We still have alanine and alanine, isoleucine, isoleucine, serine, serine, isoleucine, isoleucine, leucine, valine and valine. And now we have, instead of having leucine, we have tryptophan. And then we have, ASP. And then we have threonine. And then we have leucine. Then we have isoleucine again. This whole sequence here changed. And I kind of ran out of Legos for this, but this could be very destructive. Very destructive. But we don't know for sure until we see what happens. So the big thing here to kind of take away from this little mini lesson here is seeing how, again, it kind of goes into the grandma's cookie recipe that we talked about, where we have the DNA inside the house. It gets copied into messenger RNA by the process called transcription. And then the messenger RNA leaves the nucleus and it becomes something inside the cell body. It bonds to a ribosome, and then it takes the messenger RNA and it signals for our amino acids. Amino acids form chains. Once they leave the ribosome, these chains will start to fold upon each other, and they will form something. There'll be a protein or an enzyme, and it kind of folds it, and it becomes its own little unit. And it floats away, and goes and interacts with something. It interacts with something, go forth, do great things, then interact with something else again. They keep on going. But mutations is when you change one of these letters. Mutations can be good, mutations can be bad, and it depends on the type of mutation and what it does to the protein and what the protein actually does to the body, and if the mutation keeps on going, how bad it is or how good it is. There's an example here, deleting a nucleotide, that bad. The example over here, where we just changed one letter. Oh, that's not it. That's the wrong one. Oh yeah, here it is. We just changed one letter from an A to a T. It just changed the last letter of the codon. It's pretty resilient. Pretty resilient. Got a built-in safety feature there. It became the same thing. All right. So hopefully this is interesting to look at. Again, I'd love to have you guys play with the Legos in person, but just kind of a quick snapshot about mutations and looking at the building blocks of life.